Hey guys, this is Dr. Boney. This is just a review of some of the more poignant parts of Chapter 7. Um, and I'm just going to kind of get into it. The learning objectives are genetic variation and how that results in disease. There's many different ways that happens. So this chapter is going to cover um, some of the different ways, some of which you're familiar with, some of others you might not, um, and how these genetic variations can result in either loss of function, gain of function, um, disease, etc. I'm not going to go over every single slide. That's obviously your job. I'm going to go over the major points. So the first category here is really the two different categories of genetic variants that can cause disease. One of which is some sort of nucleotide that changes some sort of amino acid sequence and hence the function of the protein. The second one would be variations that change the amount of gene that's produced. That's not this slide. Indeed, many diseases are caused by a loss of function or reduced function. Perfect one would be like BRCA1, BRCA2, P53, things like that, where a mutation will decrease the efficiency or effectiveness of a protein in doing something important. In those cases, it's obviously tumor suppression. And then there's other proteins that these changes cause some sort of gain or a new function that not necessarily might not be good. Hence, for example, something like a protein that didn't bind to X, and now because of the mutation, it does bind to X and it shouldn't bind to X. Is one generic situation where that would be the case. The other category, the amount of a gene produced. Obviously, there are other factors besides just the nucleotide sequence that determines what proteins are made. There's many epigenetic control systems, and not only that, but some proteins that would dictate the relative production of transcription translation can alter the amount of a gene that's produced, or sometimes also alter the copy number, the amounts of the numbers of copies of the gene that exist, and hence affecting the amount of protein that exists, which may be bad or may be good. Usually is bad, but can be good. This is just one generalized class, PTC inducing. This is basically premature termination codon. This is when you would have a mutation that coded for UGA, UAA, or UAG, all of which do not have an associated tRNA and therefore no amino acid. In this case, the mRNA will be unstable and the protein will not be produced. And if it is produced, it'll be truncated and dysfunctional. So that's one example. Another one is changing copy number. And sometimes, first of all, we don't all have exactly the same numbers of copies of all genes. But in some cases, the numbers, if they become too high or too low, are out of the norm, and therefore can decrease or increase a product above and beyond what's considered normal. And that can have a deleterious effect on health. Also, we can have a situation where there's some regulatory sequence that's changed. And a lot of the regulatory proteins like transcription factors and things like that, or genes that affect, or that genes that transcription factors affect, could control the amount of the gene product. That can happen through the transcription part or the translation part. So if, let's just say, for example, you need X amount of a protein, I'll make one up, like insulin. Insulin, the amount of the gene is regulated in part by the amount of sugar, particularly glucose that's in the blood and also in the pancreas. And at some point, once there is a negative feedback loop, the amount of insulin decreases with the decreased amount of glucose that's present. However, if there's a regulatory gene that is aware of that in a normal sense and is dysfunctional, then you may not produce enough insulin, or you may produce too much insulin. It's not the insulin gene's fault, but it's the regulatory gene that controls the amount of insulin that's produced. It's that thing's fault. So this part, I'm assuming you had some of this in previous courses. I'm going to go over it relatively quickly because this shouldn't be new. But if it is, that's fine too. This is more so the different nucleotide substitutions and their effects. If someone says, hey, you have to have a genetic mutation, I would definitely say, sure, sounds good. Give me a silent. I'll take that all day long. Um, and of course, a silent mutation is when you have one nucleotide that is mutated in the DNA. However, it codes for the same amino acids. So frankly, who cares? Um, Definitely the least effective and has really no change in the phenotype. Then from that point, you have missense mutation, and I think there's a bit of a misunderstanding about missense. A lot of people assume just because you have a different amino acid automatically means that you have a dysfunctional protein. That is not the case. Um, in many situations, it is, such as like sickle cell anemia. You have one letter change, it causes a drastic phenotypic difference to the structure of the red blood cell. Um, but in many cases, if a missense mutation would code for an amino acid of the relatively the same structure, charge, hydrophobicity, etc., it doesn't necessarily have to affect the protein negatively. Sometimes it can do it positively by binding stronger, but generally the missense again is when you have one nucleotide, typically not the last one, it's usually the first or the second. That causes a different amino acid to be in that spot and therefore changes either the conformation or the binding of the resultant protein and may have bad effects and may not. And so this is just more of a chart of some of the ones beyond the missense. Obviously, nonsense would be an example of what we talked about earlier, the PTC, or the premature termination codon, when you have an amino acid sequence like UAC, which would code for tyrosine, and then that last C is mutated into an A, then you have UAA, and that's a stop codon. So that would obviously be a bad thing in many cases, or the one that they give you here as well. And then there's a stop loss mutation when you have what should have been a stop codon that's replaced by something else, and then you get a longer 3' end of the protein, which could have some sort of deleterious or bad effect on the resultant protein. So you can see here this makes a lot of sense because in many cases, if we look at, like, for example, like proline, no matter what you mean or which nucleotide we have at the third position, you're going to make proline. So even in a silent mutation like that, we can see here, especially because of wobble, how that would occur. So aside from that, there's other substitutions that have to do with conservatism, which really has to do with chemically similar amino acid sequences, and I talked about this a little bit before. In this particular scenario, you can see that, for example, if you had like a lysine to an arginine substitution, you may in fact not have much of a change in the actual function of the protein because they are of the same relative structure and charge. You might not. It depends on where that mutation happens. But in general, that's the case. And then obviously some of the other moving a little bit beyond the nonsense, which really is one of the worst ones, which is frame shift mutations, where you have an insertion or a deletion. They're called indels. Somewhere along the exon part of the mRNA, and then what happens is it throws the entire frame off by one, either backwards or forwards, and basically makes a bunch of gobbledygook, an amino acid sequence past that point that makes no real sense, and generally results in a completely dysfunctional protein in many cases. So you can also have other situations where sometimes in the production of mRNA, or I should really say I guess you could say construction of mRNA, you can have exon skipping or intron retention, which are two opposite things. Exon skipping is where you have a protein coding part of the mRNA that somehow gets skipped, it gets spliced out. You can also have the opposite situation where an intron that should have got spliced didn't and is included. It is important to note that and these two things would typically cause loss of function, but don't confuse these with the notion of isoforms. Isoforms are different versions of proteins based on sometimes which exons might be skipped or not. Not all of these cause a loss of function, but that happens to be situations where a protein has different forms because of different splicing mechanisms, and those that result in sometimes functional proteins. So this is a little bit different than that. So let's take a look real quick at exon skipping. You can see here I have exon 1 and 2, and for whatever reason there was a mutation right at the critical AG of the 5' end, and that became a T, and so therefore this was not recognized as the beginning of a splice. And so because of that, you spliced out the entire exon 2 and considered this entire junction one long exon, when in fact it's not. And you can see that that is definitely not good. So tandem repeats also are basically repeats of 3 nucleotides, sometimes could be 2, most are 3. And these repeats can happen in intronic areas, which don't have much of an effect, obviously, on the protein, but also can happen in exonic. And actually, these are important because we can use them for 2 different things. The first of which we can use them for is DNA profiling. This is something that would have nothing to do with necessarily whatever proteins you make, but you can see we can use this for like DNA fingerprinting, to be able to compare one person against another person, to see like kind of who would be related or crime scenes or stuff like that. So that's just a perfect example of how these would normally exist. In a disease format, though, this is Huntington's disease. Huntington is a protein in the brain, and Huntington's disease is caused by when a person has anywhere from 37 to 80 repeats of a CAG in the middle of the function of the protein. It makes the protein dysfunctional. It's an awful disease. A friend of mine passed away from it several years ago. It's very, how should I say this? I guess emotionally and morally interesting, in the sense that you could get tested for this, at any age and know that you're going to die from it. So the question is, would you want to know that you're going to die from it or not? It basically symptomatically causes loss of function, coordination, speech, and then eventually organs. Pretty nasty disease. But in this case, you can see where a triplet variant would be a bad thing. So generally an overview for it. Large introns are very prone to the intergenic deletions, which makes sense. And insertions with the gene can involve duplication of some of the exons, which causes all kinds of crazy stuff. You'll note that I definitely skipped a few slides. That doesn't mean it's not on the test. Cover those on your own. I'm just spending the ones that are of the most importance and most fundamental. So obviously moving on to other variations, you can have unequal crossing over. And this would be in prophase 1 in all likelihood. We can see here we have homologous parachromosomes there. And when you have unequal crossover, obviously you can have some sort of chromatid exchange that's not normal, and can obviously have an ill effect on the proteins. Gene conversion, another one. You can see here we're a part of the donor inceptor DNA switch. Excuse me. And can therefore obviously cause issues, especially you can see here CG and an AT. Now you have CG in the acceptor, which is also not good. This is a pretty interesting slide. This talks about 21 hydroxylase gene, which produces a basic protein called steroid 21 hydroxylase. If you have a deficiency in this, that's one of the major causes of congenital adrenal hyperplasia. And so there are mutations that we can find through DNA testing for this. But just to show you the idea, these are all of the different, or actually not all, there's more than this, all the different ways that mutations we've identified so far of this particular gene and what it can cause. And this is very typical of a lot of proteins, that there's going to be more than one identified mutation. Some of these can be familial, meaning passed down germline, and some can just be spontaneous. But a really interesting slide nonetheless. This one is hotspots for deletions in mitochondrial DNA. And these can have all kinds of other outcomes for disease as well. And so these would be areas that we would test. Cytochrome oxidase 1 and 2, these are just parts of the particular thing. You can see there's a lot of different areas of them located. So the rest of the chapter is pretty straightforward. I'm not going to spend a lot of time, probably another like 3 or 4 minutes on the rest of the chapter, even though I've gone through half of it. It is, again, pretty straightforward stuff and doesn't require as much instruction, I think, as the first part. However, if you do have any questions, obviously, on it, let me know. This is the slide in terms of chromosome abnormalities. That's why I'm not going over the whole PowerPoint. You definitely want to pay attention to this and be able to understand each of these different types of abnormalities that can be caused, some of which I'm sure you're familiar with, like trisomies in the case of Down syndrome. Others you might not know as much from, but definitely you want to spend time knowing and be able to give an example of this chart. In terms of protein structure perspectives, I'm going to go over again just a little bit of that, and you can do the rest on your own. But basically, protein misfolding can be caused by a lot of things, and a lot of them we talked about already, which would be a mutation that would result in a different amino acid, which would cause the protein to fold differently, or it could be a triplet extension, an insertion, deletion, you name it. But aberrant protein folding is very common. And Alzheimer's is one example, as well as Parkinson, too, that you've heard of, actually, where proteins aggregate together and can disrupt the flow of either neurotransmitters in the case of Parkinson's, or the way that a neuron functions in Alzheimer's. Excuse me. And then there's actual fibrous protein mutations like sickle cell, where the particular hemoglobin part of the protein folds incorrectly and causes a dysfunctional and malformed red blood cell. The last little thing I want to talk about here would be metabolic pathway blocks, where some kind of mutation occurs, typically in some sort of enzyme. You can see here KLMN. KLMN are the substrates, and the arrows would be the enzymes that would convert K to L and L to M. You get the idea. Excuse me. And in this case, you can see that Z is not activated. That substrate can be converted to Z. But in this particular case, some enzyme was mutated that converts L to M. And so because of that, their amount goes down. The amount of L goes up, and that can sometimes basically then trigger the production of Z based on the increased amount of L and the enzyme that convert L to Z. So one example in diet of this is PKU, which is phenylketonuria. It's a disease where you cannot process phenylalanine to tyrosine. A buildup of phenylalanine over time can damage your brain. Most infants, I believe, are tested for this at birth. It's very easy to avoid symptoms by just not eating foods that have a lot of phenylalanine in them. But in this case, what happens is, you can see here the PAH enzyme, if this becomes mutated, the person cannot convert phenylalanine to tyrosine. And that's a problem. And you may notice on any pack of bubble gum you have, it doesn't matter what kind. So if you have gum on hand, if you take a look at the package, I bet you it says people with PKU contains phenylalanine. Gum contains phenylalanine. So you'll see actually there's a warning for people who are PKU sufferers that they shouldn't be chewing gum, which is really interesting. So there is my review of this chapter, and again, let me know if you have any questions.